7 U.S. states, Florida, Texas, Alabama, Mississippi, Nebraska, Montana, and Indiana, have recently moved to ban the sale of this product. At the same time, federal agencies are supporting efforts to bring it to grocery stores nationwide. In this video, I'm going to explain what this technology actually does inside the body, especially its potential effects on stem cells, and why the full scientific picture isn't being widely discussed. I'm Dr. Thomas Harrison, and after years in clinical practice, I've seen firsthand how diet influences what happens inside our bodies, particularly for people over 50. What I'm sharing here is based on peer-reviewed research, including studies published in journals like Frontiers in Nutrition, work from researchers at MIT, and official legislative reviews from multiple state governments that concluded the safety evidence was still incomplete. As always, this content is for educational purposes. If you're taking medication or managing a medical condition, it's important to consult your doctor before making major dietary changes. That's not just a formality, it truly matters, because individual health situations are unique, and no video can replace personalized medical advice. This isn't about politics, it's about understanding what the science actually says, and right now, there are important questions that aren't being clearly addressed in mainstream discussions about food. By the end of this, you'll have a clearer understanding of what cultivated meat is at the cellular level, what current research says about how it's produced, why age, especially being over 50, may play a role in how your body responds, and what alternatives may help support your body's natural regenerative systems. At the core of this debate is a simple but critical question the industry hasn't fully answered. What happens when the human body regularly consumes cells designed to keep dividing indefinitely, not just short-term but over decades? The honest answer is that we don't yet know. Long-term studies tracking people over 10 or 20 years simply haven't been completed, and yet approval for consumption has moved forward without that data. That alone is worth considering carefully. If you find this kind of deep dive helpful, subscribing helps support more work like this. No pressure, just something to keep in mind. Let's start with the science itself, because to understand the concern, you first need to understand what cultivated meat really is. This isn't plant-based food. It's not soy protein or fungus-derived alternatives. Cultivated meat, also known as lab-grown or cell-cultured meat, is created by taking living cells from animals, often muscle or fat stem cells, and placing them into controlled environments called bioreactors. Inside these systems, the cells are fed a nutrient-rich solution containing amino acids, glucose, vitamins, minerals, and growth factors. These signals encourage the cells to divide, multiply, and form tissue that resembles conventional meat. The ultimate goal is to scale this process, producing large quantities efficiently and cheaply enough to compete with traditional meat in supermarkets. On the surface, it sounds like a promising solution: less reliance on livestock, reduced environmental impact, and scalable protein production. It's easy to see why it has attracted major investment and public interest. But the concern isn't about the goal, it's about the process. Specifically, what needs to happen to these cells to make them grow rapidly enough for industrial production, and how that process might affect their biological characteristics along the way. Here's the biological reality that rarely shows up in press releases or investor presentations. When you take normal muscle cells from a chicken or a cow and grow them in a lab, they don't behave endlessly, they follow strict biological rules. Every healthy cell has a built-in limit on how many times it can divide. This is known as the Hayflick limit, a natural safeguard that prevents uncontrolled growth. That limit exists for a reason. Cells that divide without stopping are not considered normal, they're considered abnormal. In medicine, when cells lose this control and begin multiplying indefinitely, we call that process malignant transformation, which is essentially how cancer begins. For cultivated meat to be produced at large, industrial scale, companies need cells that bypass this natural limit. They need cells that can keep dividing continuously, day after day without stopping. To achieve that, the cells are either intentionally modified or cultured for long periods until they become what scientists call immortalized cell lines, cells that no longer obey normal growth limits. That alone raises an important question: what kind of cells are actually being used in this food system? A 2024 peer-reviewed paper published in Frontiers in Nutrition by researchers Hauser, Searman, Rack, and Nachman looked directly at this issue. Their findings were clear. If you want continuous large-scale production, you have to override the cell's natural controls on growth. One way to do that involves altering genes that regulate the cell cycle, specifically genes like TP53 and PTN. To understand why this matters, you need to know what these genes actually do. TP53 is one of the most important tumor suppressor genes ever identified, It's often called the guardian of the genome, and for good reason. When DNA damage occurs, whether from environmental stress, radiation, or normal replication errors, TP53 steps in. It either pauses the cell so the damage can be repaired, or triggers programmed cell death to prevent that damaged cell from continuing to divide. This gene is one of your body's primary defenses against cancer. In fact, mutations in TP53 are found in over half of all human cancers. PTN plays a similar protective role. It helps regulate cell growth and survival, keeping cell division under control. When PTN function is reduced or lost, cells receive constant signals to grow and divide, often through pathways like PI3K that drive proliferation. The paper states this plainly. One approach to scaling cultivated meat involves altering genes commonly disrupted in cancer, including TP53 and PTN. And here's the part that deserves special attention. The researchers note that these changes don't always have to be intentionally engineered. Simply keeping cells in culture for extended periods can lead to these mutations spontaneously. In other words, the very conditions required for large-scale production can create pressure that favors cells which have already begun to lose their natural growth limits. A separate 2023 study published in Frontiers in Nutrition looked at genetic drift and stem cell behavior under laboratory conditions, and its findings raised serious concerns that arguably deserved far more regulatory attention. The researchers observed that normal cells taken from non-primate animals, the same types commonly used in cultivated meat, began developing what they described as a tumor-like profile after just 2 passages in culture. That means after only 2 cycles of cell division in a lab environment. These weren't abnormal cells to begin with. They started as healthy functional tissue. But within a very short time in culture, they began showing genetic traits associated with tumor behavior, including signs linked to potential metastasis. Because of this, the authors recommended something very specific: mandatory genetic quality control testing for all cell lines used in cultivated meat production, with a particular focus on detecting mutations that activate oncogenes. As of now, that recommendation has not been made a requirement by the FDA or other major food safety authorities. Now, to be fair, and accuracy matters here, the cultivated meat industry makes a point that is technically valid within its scope. They argue that consuming cells with altered tumor suppressor genes is not the same as eating cancer. The human digestive system is not passive. Stomach acid breaks down proteins, and enzymes degrade DNA into smaller fragments. The industry's position is based on the assumption that any genetic material from these cells is fully broken down during digestion and does not remain intact long enough to have biological effect in the body. That's their stated safety argument, and it shouldn't be dismissed outright. But it leads to a much bigger, still unresolved question. What evidence supports that assumption over the long term? Where are the studies tracking people who have consumed these products daily for 10 or 20 years? What population has been followed long enough to measure real outcomes? This gap has been acknowledged in the scientific literature. Researchers at MIT addressed it directly in a widely cited 2021 paper, noting that confirming the long-term safety of products made from immortalized animal cells, whether through spontaneous changes or genetic engineering, remains an unresolved area of research. In other words, the safety question is not fully settled. When the FDA approved products from companies like Upside Foods and Good Meat in 2023, it did so through a process called a cell culture consultation. This involves reviewing safety data submitted by the companies themselves. It does not include long-term human trials, decade-long follow-ups, or large-scale population studies. For context, Singapore became the first country to approve cultivated meat back in 2020. Even there, the longest real-world consumption window is still under 5 years. That's simply not enough time or scale to detect long-term health outcomes, especially subtle or slow-developing ones. There isn't yet a robust epidemiological system in place to monitor these effects over time. One way to think about this situation is through an analogy. Imagine you're about to board a plane. You ask if it's safe. The engineers tell you it has passed all pre-flight inspections, and that based on theory and available data, it should operate correctly. But they also admit that this particular aircraft design has never been tested beyond a few years of use, that some of its key components were engineered to bypass traditional safety limits, and that full details of those modifications aren't publicly available because they're proprietary. You'd probably hesitate. And yet in this case, not only has approval been granted, the product is actively being promoted for widespread consumption, even as some of the most important long-term questions remain unanswered. Let's move into what may be the most important layer of this entire discussion, especially if you're in your 50s, 60s, or beyond. This isn't a one-size-fits-all issue. The biological context actually changes with age, and that matters more than most people realize. Your stem cells today carry a kind of biological history, a record of every time they've divided throughout your life. Each division, whether it's repairing tissue, maintaining organs, or responding to stress, carries a small chance of error. Most of those errors are corrected by your body's repair systems, but not all of them. Over time, a small number of mutations accumulate. That's not disease, that's simply how normal aging works at the cellular level. As you get older, two things happen. Your baseline level of accumulated mutations increases. Your body's ability to detect and repair DNA damage becomes less efficient. Even systems like TP53, often called the body's genetic guardian, don't function as strongly as they once did. This gradual decline is one of the key reasons why cancer risk rises significantly in later decades of life. Now consider that context. If a person over 50 is regularly consuming a product created through processes that involve altering or bypassing natural growth controls, like tumor suppressor pathways, the question becomes, does that interaction matter biologically? The honest answer is that we don't know, because that specific question hasn't been directly studied. No long-term trials, no targeted research on older populations, no data tracking outcomes over decades. And it's important to be clear, the absence of evidence isn't proof of safety, it simply means the question hasn't been fully explored. Ironically, the group most likely to think carefully about diet, people over 50 who are actively trying to support their health, is also the group for whom this gap in evidence may be most relevant. There's another biological factor that deserves attention here: growth factor signaling. To keep cells alive and multiplying in bioreactors for large-scale production, cultivated meat relies on continuous exposure to growth signals. These include compounds like IGF-1, insulin-like growth factor 1, fibroblast growth factor (FGF), epidermal growth factor (EGF). These molecules tell cells to grow, divide, and survive. In the human body, these signals are tightly controlled. They rise when needed, during healing, exercise, or growth, and then drop back down once the job is done. This balanced on and off signaling is essential for normal physiology, but in industrial cell culture, these growth signals are often present continuously, at levels designed to support ongoing cell proliferation. That raises an important question: do any of these signals or their effects carry over into the final product? And if so, does regular consumption influence the body over time? Again, this hasn't been fully answered, and this is where age becomes especially relevant. In longevity and cancer research, IGF-1 levels have been studied extensively, particularly in older adults. Across multiple large studies and meta-analyses, including work published in journals like The Lancet Oncology, higher circulating IGF-1 levels have been consistently associated with increased risks of certain cancers, including prostate cancer, breast cancer, colorectal cancer. Importantly, these associations tend to be stronger in people over 50. That doesn't automatically mean cause and effect, but it does highlight a well-established biological relationship that researchers take seriously. So when you combine age-related changes in DNA repair, increased baseline mutation load, and exposure to systems involving sustained growth signaling, you arrive at a set of questions that are scientifically important and still largely unanswered. The biological mechanism here is actually well understood. IGF-1 activates a pathway inside cells known as PI3K, AKT-mTOR. This pathway promotes cell survival and growth, while at the same time reducing apoptosis, the natural process your body uses to eliminate damaged or potentially dangerous cells. In a younger body, this system is tightly balanced. But with age, when tumor suppressor systems are already less efficient, persistent activation of this pathway can create conditions where abnormal cells are more likely to survive instead of being cleared. That sequence enhanced survival signaling combined with reduced cellular cleanup is one of the key biological patterns involved in the development of malignancy. Now I want to be absolutely clear about something. I am not claiming that cultivated meat causes cancer. There is no direct evidence supporting that claim, and it would be irresponsible to say otherwise. What can be said based on published research is this: the processes used to produce cultivated meat at scale involve genetic and molecular pathways that are well known in cancer biology, and there is no long-term human data tracking the effects of regular consumption over many years or decades. Both of those statements are factual and supported by current literature. Taken together, they don't prove harm, but they do justify careful ongoing scrutiny rather than assuming safety based solely on short-term evaluations and theoretical models. Now let's look at a third factor, one that often gets less attention, but may be more immediately relevant to how your body actually feels and functions: Whole animal foods, especially from grass-fed, pasture-raised sources, are not just protein, they're biologically complex. They naturally contain a diverse microbial presence that has co-evolved with human digestion, enzymes that assist in breaking down food, connective tissue rich in amino acids like glycine, proline and hydroxyproline, which support joints, gut lining and tissue repair, fats that include compounds like CLA, conjugated linoleic acid, studied for roles in inflammation regulation and metabolic health, nutrients like vitamin K2, MK4, which helps direct calcium into bones and away from arteries, something especially relevant as vascular health becomes more important with age. Cultivated meat, by contrast, is produced in a sterile, controlled environment. It may replicate the structure of muscle tissue, but it lacks much of the broader biological context found in traditional animal foods. In that sense, it's closer to an isolated component than a fully integrated food system. For someone over 50, this difference may matter more because gut microbiome diversity tends to decline, digestive enzyme production decreases, absorption of certain nutrients becomes less efficient. So replacing a food your biology has adapted to over thousands of years with something entirely new without long-term data is not necessarily a neutral swap. The fourth area of concern comes from a different angle entirely: microplastics. This issue was raised directly during legislative discussions in places like Texas. Cultivated meat is grown inside industrial bioreactors, which are typically made with plastic components, tubing, vessel linings, and sometimes scaffolding materials that help cells form structured tissue. That means the cells are in constant contact with synthetic materials during the production process. Some policymakers have pointed out the possibility that this could introduce microplastic exposure or affect cellular integrity during growth. This isn't a fringe idea. Microplastics are now a major focus in environmental and health research, with growing evidence that they can interact with biological systems in ways we're still working to fully understand. Stepping back, none of these points on their own prove harm, but together they highlight something important. This is a new technology entering the food system, involving biological processes that are complex, not fully studied over the long term, and potentially more relevant for certain populations, especially older adults. That doesn't call for panic, but it does call for informed caution and continued investigation. Over the past few years, research on microplastics has grown rapidly, and the findings are becoming harder to ignore. Scientists have now detected microplastic particles and related compounds in multiple parts of the human body, including blood samples, lung tissue, arterial plaque, even placental tissue. At the same time, researchers are actively studying how these particles interact with cell membranes and biological systems. It's a fast-evolving field, but one thing is clear: microplastics are no longer just an environmental issue, they're a biological one. Now place that into the context of cultivated meat production. This is a system where living cells, the very material that becomes the final food, are grown in continuous contact with plastic-based equipment. That includes reactor walls, tubing, and in some cases scaffold structures that help shape the tissue. These interactions occur over extended periods, often under conditions like elevated temperatures and constant exposure to growth signals. And yet there is currently no publicly available peer-reviewed research clearly characterizing whether or how microplastic material might be incorporated into the final product. Now let's step back and look at the financial side of this industry, because it helps explain how and why things are moving so quickly. Major players are already deeply involved. Bill Gates is one of the largest individual investors in cultivated meat ventures. Tyson Foods, one of the biggest meat producers in the US, has invested in companies like Upside Foods. JBS, the largest beef processor globally, has also entered the space through acquisitions. These aren't small startups disrupting the system from the outside. They are the same dominant forces in global food production, positioning themselves to lead whatever comes next. From a business perspective, their incentives are clear: move fast, secure regulatory approval, and scale commercially. Waiting decades for long-term safety data doesn't align with that model. Then there's the issue of transparency and intellectual property, which is where things become even more complex. Some of the leading companies in this field have chosen not to patent their core production methods. At first glance, that might not seem unusual, but it has important implications. Filing a patent requires full public disclosure. That means every step of the production process, any genetic modifications, the composition of growth media, all chemical and biological inputs, all of it becomes part of the public record, available for regulators, scientists, and independent reviewers to examine. By choosing instead to protect their processes as trade secrets, these companies avoid that level of disclosure. The result? The exact methods used to grow and modify the cells in these products remain largely hidden from public view. A 2024 paper in Frontiers in Nutrition highlighted this pattern directly, noting that companies are intentionally structuring their intellectual property strategies to limit transparency. In most areas of food safety, that level of opacity would raise serious concerns. For a completely new category of food, it would typically trigger demands for more, not less, disclosure. Yet in this case, it has largely been accepted as standard practice. Finally, it's worth looking at how this issue has played out at the state level. When Florida passed legislation restricting cultivated meat, the public messaging included political language that made it easy for critics to dismiss the move as rhetoric rather than policy. But if you set aside the politics and focus purely on the underlying action, a different picture emerges. 7 separate state governments, each reviewing available scientific evidence independently, arrived at a similar conclusion. The current safety data was not sufficient to justify widespread commercial sale. That doesn't automatically mean the product is unsafe, but it does mean that multiple independent bodies looked at the same evidence and decided that more certainty was needed before moving forward. Regardless of how you feel about the politicians involved, the core regulatory decision itself is real, documented, and based on a clear issue: the lack of long-term safety data. The same gap acknowledged by MIT researchers in published literature. 7 states looked at that gap and decided it was reason enough to pause before allowing widespread commercial sale. The federal government reviewed the same landscape and reached a different conclusion. Now let's shift to what actually matters most: what you can do with this information. Because at the end of the day, decisions about food aren't theoretical, they're daily, practical choices. And when it comes to supporting your biology, especially after 50, the evidence strongly favors dietary patterns that humans have relied on for generations and that modern research continues to support. 1. Grass-fed beef, not grain-fed. Beef from animals raised entirely on pasture has a meaningfully different nutritional profile compared to conventional grain-fed beef. It contains 2-5 times more CLA, conjugated linoleic acid. Depending on season and forage quality, CLA has been studied for its role in supporting anti-inflammatory pathways and cellular regulation. It provides vitamin K2, MK4 in its fat, important for directing calcium into bones and away from arteries, which becomes increasingly relevant with age. Practical guidance: aim for about 85-140 grams per serving, 3-4 times per week. And importantly, don't remove the fat, because that's where many of these nutrients are concentrated. 2. Bone broth for structural support. Bone broth made from collagen-rich parts like joints, marrow bones or chicken feet releases key amino acids into the liquid: glycine, proline, hydroxyproline. These aren't just structural, they also act as signals that support joint and cartilage health, bone density, tendon strength. Gut lining integrity research, especially in aging models, shows glycine plays a role in tissue repair and gut barrier function. How to use it: drink 1-2 cups daily, ideally before your first meal. Add about 1 tablespoon of apple cider vinegar at the start of cooking to help extract minerals. Simmer 12 hours for poultry bones, 24 hours for beef bones. 3. Beef liver, once a week. This is the one many people hesitate about, but nutritionally it's hard to match. Beef liver is one of the richest natural sources of CoQ10, important for cellular energy. The most bioavailable source of vitamin B12, high in copper and zinc, informs your body absorbs efficiently. For people over 50, these nutrients are often low and harder to absorb from supplements alone. Why B12 matters here: vitamin B12 is essential for DNA synthesis, especially in rapidly dividing cells. That makes it critical for stem cell function, particularly in the blood and immune system. The bigger picture: the goal isn't fear, it's informed choice. When long-term data is limited, one reasonable approach is to lean toward foods that have a long history of human consumption, are supported by both traditional use and modern research, provide complete, biologically active nutrition. Especially as your body changes with age, those choices tend to matter more, not less. The stem cells in your bone marrow are responsible for producing every red blood cell, white blood cell, and platelet in your body. For these cells to function properly, they rely on efficient DNA synthesis, and that process depends heavily on vitamin B12. When B12 levels are too low, DNA production becomes impaired. This leads to what's known as megaloblastic changes, where blood cells are formed incorrectly. Clinically, this reflects a breakdown in normal bone marrow stem cell function. And this isn't a rare issue. Studies consistently show that 30–40% of people over 50 have some level of B12 deficiency. This happens partly because stomach acid needed to release B12 from food declines with age. Intrinsic factor required for absorption also decreases. Just 100 grams of beef liver once per week can provide more than 400% of your daily B12 needs, along with high levels of copper, zinc, and vitamin A. If the taste is a challenge, there's a simple fix. Soak the liver in whole milk for 2-4 hours before cooking. This helps reduce the strong flavor significantly. Small fatty fish, 3-4 times per week. Fish like sardines, mackerel, anchovies, and herring are incredibly valuable, especially when you eat them with their soft, edible bones. They provide omega-3 fatty acids, EPA and DHA, which help reduce inflammation in the bone marrow environment where stem cells live. Support for resolving chronic low-grade inflammation. A key driver of aging, the soft bones in fish like canned sardines are rich in calcium and phosphorus, in highly absorbable forms that support bone strength. They also contain vitamin D3, which helps guide stem cells toward forming bone-building cells, osteoblasts, rather than fat-storing cells, a balance that tends to shift unfavorably with age. Canned sardines are just as effective nutritionally as fresh ones, and the bones are completely safe and beneficial to eat. Pasture-raised whole eggs, 2-3 daily. Eggs are one of the best sources of choline, a nutrient many people over 50 don't get enough of. Choline is essential for DNA methylation, a key epigenetic process, building healthy cell membranes, producing acetylcholine, a neurotransmitter important for brain function. When choline intake is low, it can disrupt the epigenetic signals that help stem cells maintain their identity and function over time. DNA methylation, in particular, is one of the core mechanisms cells use to regulate gene activity, and disruptions in this process are strongly linked to aging. As for cholesterol concerns, more recent large-scale studies have shown that egg consumption is not strongly linked to cardiovascular risk in otherwise healthy individuals. Earlier concerns were largely influenced by broader dietary patterns, not eggs themselves. Fermented foods, daily, small amounts, foods like raw sauerkraut, kimchi, traditional whole milk yogurt, provide beneficial bacteria such as lactobacillus and bifidobacterium. These microbes help produce butyrate, a short-chain fatty acid created during fiber fermentation in the gut. Butyrate plays a powerful role in supporting gut health, regulating gene expression through epigenetic mechanisms. It works by inhibiting enzymes, histone deacetylases, that when overactive, can disrupt normal cellular behavior, something seen in both aging tissues and early disease processes. A simple habit: add 2-3 tablespoons with meals. Wild blueberries, 3-4 times per week. If available, aim for about half a cup, fresh or frozen. Wild blueberries contain higher levels of partyrostilbane anthocyanins. These compounds activate SIRAT1, a protein involved in regulating cellular aging and stem cell behavior. One of SIRAT1's roles is maintaining stem cell quiescence, a resting state where cells are preserved and protected, rather than constantly dividing. This state isn't inactivity, it's a controlled protective mode that helps maintain regenerative capacity over time. Putting it all together, these foods don't work through a single pathway, they support multiple systems at once: DNA repair and synthesis, inflammation control, epigenetic stability, stem cell preservation and function, and importantly, they come from dietary patterns your body has adapted to over thousands of years, supported now by a growing body of modern research. For someone over 50, that combination of biological familiarity and scientific support is a strong foundation for making daily food choices. When stem cells are pushed to divide constantly, they can burn through their regenerative capacity faster than the body can replace them. Over time, that means a smaller reserve available for repair and maintenance later in life. By contrast, signals that support proper quiescence, a controlled resting state, help preserve that reserve so your body can continue maintaining tissues well into your 70s, 80s, and beyond. From years of clinical experience working with people in this age group, one pattern stands out clearly: the people who see the most lasting results are not the ones who try to overhaul everything at once. They're the ones who choose 3 realistic dietary changes, follow them consistently for 30 days, then gradually build from there. That approach works because your biology doesn't change overnight. Your gut microbiome, your mitochondrial function, your inflammatory balance, all respond to sustained input, but on a timeline of weeks to months, not days. Expecting dramatic results in the first week and giving up when they don't happen is one of the most common reasons people fail to see progress. Important precautions before making changes. If you're considering significant dietary adjustments, a few situations require extra care. Blood thinners, especially warfarin, increasing vitamin K intake from foods like liver, eggs, or fermented vegetables can affect clotting levels and may require dose adjustments. Chronic kidney disease stage 3 or 4, higher protein and phosphorus intake needs to be carefully managed with your doctor. Gout or high uric acid, foods like organ meats and certain fish may need to be moderated. Active cancer treatment, any dietary changes should be discussed directly with your oncologist. What to realistically expect, based on both research and clinical observation. Within 2 weeks, more stable energy throughout the day, less afternoon fatigue. This reflects improved energy production at the cellular level. Within 4-6 weeks, better skin texture, improved hair quality, faster recovery after physical activity. These are signs of improved function in fast-renewing tissues. At 3-4 months, noticeable improvements in inflammation markers on blood tests, increased strength like grip strength, better mental clarity and sustained focus. These longer-term changes matter most, and they take time because stem cell turnover in most tissues happens on a monthly, not weekly cycle. The bigger picture, the changes are real, but they require patience and consistency. And stepping back, this conversation points to something broader. Our food system is evolving rapidly, often faster than the science needed to fully evaluate those changes before they reach the public at scale. We've seen this before. For example, trans fats were once introduced as a healthier alternative to traditional fats. They remained widely used for decades before strong evidence revealed their link to cardiovascular disease. That history doesn't mean every new technology is harmful, but it does highlight a recurring pattern. Adoption can move faster than understanding. And when that happens, the most reliable strategy is not fear, but informed, thoughtful decision-making, grounded in both evidence and long-term perspective. High-fructose corn syrup was rolled out on a massive scale after short-term studies suggested it behaved similarly to other sugars, long before we fully understood its long-term effects on liver fat, uric acid balance, and metabolic health. Industrial seed oils followed a similar path. They replaced traditional animal fats across much of the food supply, based on early hypotheses about saturated fat and heart disease, hypotheses that in hindsight were influenced as much by institutional momentum and economic incentives as by strong conclusive evidence. In each of these cases, innovation moved faster than validation, and over time large populations lived with the consequences before the science caught up. Cultivated meat may ultimately prove to be completely safe for long-term use. That's a real possibility, but the honest answer today is this: we don't know yet. And neither does the scientific community, because the long-term data required to answer that question simply doesn't exist. What is known from current research is that the production process involves biological pathways like TP53 regulation, growth signaling, and cell immortalization that are deeply studied in cancer biology. The companies leading this space have largely chosen trade secrecy over full transparency, limiting independent review. Multiple state governments reviewed the available evidence and concluded that the safety case wasn't yet complete. At the same time, the group most likely to think carefully about diet, people over 50, is also the group with higher accumulated cellular mutations, reduced DNA repair efficiency, stronger links between growth signaling and disease risk. For them, the absence of long-term safety data isn't theoretical, it's personal. The federal government approved it. 7 states chose to hold back. You can decide for yourself what those choices reflect. What you control, no matter what, is what ends up on your plate. Foods like grass-fed beef, bone broth, liver sardines, eggs, fermented vegetables, blueberries. These aren't new inventions or complicated protocols. They're foods humans have relied on for generations, now supported by modern research showing they help support stem cell function, reduce chronic inflammation, maintain stable gene expression, build resilience as the body ages. They work with your biology, not against it. They don't depend on bypassing natural safeguards. Here's the clearest summary of where things stand today. Cultivated meat has not been proven harmful. It has also not been proven safe over decades of use. Its production involves biological mechanisms that are still under active study. Full transparency around how it's made is limited. Long-term human trials have not been completed. Some governments have chosen to wait before approving it. Those are the facts as they currently exist. Keep asking questions, especially the ones that aren't answered in marketing materials. That habit will serve you far better than any single diet change. If this raised something specific for you, think about it, question it, and keep exploring. And if you want to dive deeper into how to support your body's regenerative systems as you age, the nutrition, the biology, and realistic expectations, you can continue building on that knowledge, step by step. Stay curious,